My name is David Simons. I'm a PhD student at the Royal Veterinary College in London in the UK, and I conduct my research on emerging zoonotic infectious diseases, predominantly Lassa fever in West Africa. So I'm going to present the process and sort of the findings of our recent pilot study conducted in November and December of last year. So Lassa fever is a viral hemorrhagic fever endemic in several countries in West Africa, predominantly Nigeria, Sierra Leone, Liberia, and Guinea. It is found in its reservoir species, Mastitis natalensis, and then it spills over into human populations. And these spillovers come in sort of epidemic waves, typically in sort of January to March of the year, and that's believed to be associated with increased rainfall. However, there's possibly further factors that can increase the risk of these spillover events, such as land use change or rodent assemblages and other sort of species that are competing and controlling Mastitis natalensis populations. So that's what my work is going to be focusing on, and I'm trying to develop a model to look at the different rodent species and how their populations vary. So what we did is, in order to identify where we wanted to trap the rodents and where we wanted to investigate the populations, is that we looked at previous Lassa fever cases from a review by Gibb in 2017. And we use recent IUCN habitat classifications based on satellite images from Jung et al.'s paper in 2020. And we drew a buffer zone around each previous case. And that gave us the type of land use that sort of cases are associated with in a rough manner. So following that, we identified two villages in eastern Sierra Leone, and they were Salama and Lalihan. And we went to those villages and met with the communities who were happy for us to sort of conduct our trapping activity there. So we set up our traps in 5 different sites to cover a good range of those different habitats. And we obtained a trap success rate of around 3 to 3.8%, which is fairly common in rodent trapping studies. So the main sort of outcomes from this research is that we found a suitable method, data collection tools. We had villages that had good buy-in to our study. And we identified that we'd conducted a fairly comprehensive survey of the small mammal populations there. We needed to slightly increase the number of trap nights we performed in Salama, but in Lalihan we reached sort of an asymptote on our species accumulation curves. So we will continue using these villages going forwards into our longitudinal study, and we will have repeat trapping activities in these villages and several others in order to better understand the rodent population dynamics in eastern Sierra Leone. If you want to read further about this work on the associated QR link, will take you to my webpage where I've written this up with quite a lot more information and detail on the individual rodents that we trapped. And I'd be happy to answer any questions as well.